Chapter 1, Section 14, Molecular Basis of Life. Now, a lot of what we know about how cells work, how DNA polymerases work, how ribosomes work, how things get transported across membranes. Those things that we know, we didn't learn from studying humans. No, because our cells are too complex. Our cells have way too much going on. Our cells are also really hard to culture. Same with other animal cells, same with plant cells. But bacteria are stupid easy. Hardest part about them is controlling what can grow and under what conditions and making sure we maintain a pure culture. Because just a stray, dust mote with a couple of fungal or bacterial spores gets down into whatever you're working on, and they can overgrow. But because bacteria are so easy to grow, so quickly to grow, and they're rudimentary compared to some of our metabolic processes, they've been great at studying everything. How certain enzymes work, how DNA works, what does it take to get, certain genes expressed, turned on, turned off, all of that stuff. So what we know about modern day genetics, what we know about modern day cell biology, well, a lot of that is because of bacteria. Now, yes, we start at bacteria. Okay, bacteria are not like us. Okay, comparing some of our enzymes and some of our processes to bacteria is like comparing dial-up to fiber optic high-speed internet access. Okay, that's about what it's like. Okay, dial-up works and it'll get the job done, but it's not as fast, it's not as efficient, and it's not as foolproof as the fiber optic high-speed blah 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 blah. We start with these guys to get an idea of how things work, and then it cuts down on how much stuff we have to wade through to try to figure out in eukaryotes, plant, animal cells, and whatnot. I mean, you take a look at some of the people, you've heard these names before. I'm not going to go into what they all did. Griffith, Avery, McLeod, McCarthy, Watson, Crick, Franklin. Zuckerandl and Pauling, Linus Pauling, they came up with the idea, the molecular sequences, evolutionary sequence relationship, how the sequences came up to equal, certain amino acid sequences, certain polypeptides. This is the DNA, AGCT. This is how it fits to the amino acid sequence. This is how the amino acid sequence fits to the polypeptide, the protein, and whatnot. Here they're just showing, the work. You remember this, the R, the S, this is all based on Streptococcus pneumoniae. R has a rough appearance colonies, S had a smooth because they had a glycocalyx, which we'll talk about in a few chapters. One's a killer, the other's not. Go through. Griffith showed, hey, look, if I take R and killed S, the killer, I boiled the S till it's killed, I mix them together, something transfers from the dead S to the living R, and I end up with killer S, even though I started off with only living R. Avery, McLeod, McCarthy went and repeated this and showed, hey, look, if we add DNAs, N's and A's means it's an enzyme, DNAs means it's an enzyme that cuts up DNA. Well, this gets stopped. RNAs, proteases don't stop it, but it's only when I interfere with DNA that this Mickey Mouse here doesn't die.